fucking insane amount of work.
Yep.
By many, 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 many people.
And, and then the amount of data that had to be collected now. And so here's the difference is that, there's data, there's evidence, there's conclusions and proof. And that's an uphill climb. But proof, the next one up is meaning. My lab has been largely responsible, at least partly responsible, for the data deluge that's out there in the world, both in how to do tissue biopsy analysis, how to do single-cell analysis, et cetera. And, data felt good for a while. It was like this, this feedback loop of, oh, wow, I can get all this data. And then suddenly you look at it and you go, well, what the fuck does it mean? And so humanity has this habit of backing itself into a corner and then suddenly finding this eureka moment that gets it out. And so our eureka moment about 2 years ago was artificial intelligence, where suddenly I had the ability. So normally I would collect all this data and go, okay, well, it seems myeloid suppressor cells are important here and T regulatory cells are important here. Okay, I get on the phone or I send an email to whoever the local expert is, either on Stanford campus or around the world, and try to get some information from them. But then now you're dealing with hundreds of cell types, each individually of which have thousands of variations themselves. And each subtle variation means something. And there's no expert for any of that. But AI can be, at least in part, that expert. So suddenly I have 22 million papers published, in all the fields of science, several tens of millions just in, several millions just in immunology alone. And AI can be the sleuth for me, can be both the angel and the devil on my shoulder that can make sense of things in ways that I never would have been able to before, especially with agentic AI. So we, for instance, in my lab, have developed an agentic AI that is basically an immunologist scientist in a box. We can give it the raw data, and we can pose a question in natural language, and then we say, hey, make sense of this and turn it into a network. Normally, that would have taken a graduate student along with a couple of postdocs months and months and months to put it all together. Now, in 3 hours, we can get pictures and hypotheses of how all that data fits together in ways that I never could have done before. At the beginning, it did a lot of hallucinations, which you probably heard about in AI. But my answer to my colleagues is some of my best students hallucinate. Right. And so, but the human's still in the loop. And so with all of this together, now we can make meaning out of the data. And we can skip a lot of the intermediary steps and speed it up. And it's just getting better. I mean, we, for instance, have put in a couple of papers now where, so for instance, where one of my recent specialties is what's called the tumor-immune interface. So you have the tumor, you have the immune system, which is coalescing on, near, and then in some cases the tumor creates a boundary, a barrier between itself and the immune system, where there might be certain kinds of cells that the immune system, the tumor has told the immune system, ignore us, we're not here. But what we now can do is on the other side of when you look at, let's say, complex patient populations, you find these things called tertiary lymphoid structures. So your body has 220 or so lymph nodes, okay? And the lymph nodes are where the immune system makes decisions, let's say. It turns out that in the middle of tumors, the body has evolved a mechanism to create what essentially looks like a lymphoid structure in the middle of the tumor. It's sort of a forward camp of immune cells that the more of those you see in a tumor, the better will be your outcome as a patient. And so we used a cohort of colorectal cell, basically colon cancer patients, where we looked at hundreds of biopsies. And we did that pseudotime analysis where we looked for mature tertiary lymphoid structures. And then we looked for immature, slightly less mature, even more less mature, et cetera. And we were able to backtrack to the cell types which need to come together that would then form the more mature. What use is that? It's a nice paper. But it also now tells us what we might do to create more of these in a tumor. Because the more we already know from multiple kinds of tumor types now that the more of these tertiary lymphoid structures you have, the better off will be your outcome with chemotherapy. So it might be, for instance, that once we know that you have a disease like this, we could give you some kind of therapy, a virus or what have you, that goes and homes to the tumor, seeds the beginnings of these initiators there's these cytokines that are produced that are necessary for initiating the formation of these objects. And so there's a huge benefit to that, but we never would have found those, in my lab at least, without the AI. Because it basically did the work for us.
That's fascinating.
Yeah.
Now, are you using like a standard large language model, or do you have like a specific structure that's built that interfaces with large language?
Correct. So we use, we can use pretty much any of the LLMs, but right now we find that OpenAI is the best, for us at least. And then we create an agentic overlay. Basically what's called, you probably know, chain of thought, which is a series of questions. So how we taught it was we basically came up, here's 100 kinds of questions a scientist would ask about the immune system. And then we tell ChatGPT, now create 1,000 questions like this. So, it's artificial data or artificial questions. We curate those to make sure that they're good. Then we do 100 hypotheses, and we create thousands of types of hypotheses, et cetera, and the same for tests that you might run. So now, from A to Z, we have an agentic AI that you give it raw data, it knows what to do with the data, it then generates hypotheses for you, and then it literally tells you the kinds of experiments you should do next to prove or disprove the hypothesis from the raw data.
It's a genius in the lab with you.
Exactly.
Is OpenAI learning from this agentic AI?
Oh, yeah.
So there's a mutually beneficial relationship.
Yeah, I mean, we're not working with them directly on it.
But you use it, and because you
use
use it with your AI,
right,
it's benefiting from it.
And we first thought to turn it into a company, because that's kind of one of the things we do in my lab, because I've always thought that it's important to give back to the taxpayer the money that they've invested in us. And the best way to do that is commercialization. I'm totally unapologetic about that, even though that got me in a lot of trouble at Stanford in the early days when, making money was, commercialization was evil, and even at Stanford. And so I think that that's an important process because scientists are good at asking maybe the questions and coming up with solutions, but scientists aren't the best at commercializing it and turning it into a product that can be used or testing it, in large communities. So the AI that we developed, we thought, okay, well, maybe we can do this. But we thought, you know what? AI is moving so fast. Why don't we just give this to the community? Why don't we open source this? We can use it for maybe specific targeted purposes, but we're basically going to publish the whole thing on GitHub. To let other people use it. Because we've seen other people make claims about stuff that they've already made, and it's like, ours is better. So why don't we just put it on GitHub and let people learn from it? The resistance to the commercialization, what was the initial argument? So back when I was a grad student in the 80s, basic research, as opposed to translational research, was considered the height of intellectual desire, right? Basic research, and we're not here to make money, we're here to discover things. And that's important. And nearly every major discovery and every major therapy in the world came from basic research. But then, there were limits to how much money you could give to basic research, and then there was a desire at a certain point to say, hey, well, are you going to do anything about this? Are you going to make it? And so translational research became a push. So there's a guy at Stanford by the name of Paul Berg, who won the Nobel Prize for recombinant DNA way back in the day, and Paul came up with this concept, bench to bedside. Meaning that we don't have to be either or. We can be part of an arc, and Stanford wanted to be and enable within the medical school both the basic research, which we were great at, as well as bringing it directly to the patients as well. So to link clinicians and the desires of clinicians with the basic researchers. I mean, most scientists would be happy just to study anything. Just point me at something and I'll be happy if I can get interested in it. So, and we're no more happy than when somebody recognizes the value of what we do.
Right.
But basic research was sort of the height, and there was a push against anybody trying to commercialize. So when I started as an assistant professor, so I started as a grad student. I went to MIT to work with this guy, David Baltimore, who won the Nobel for reverse transcriptase. And then I wanted to come straight back to Stanford because I already felt that it was a positive environment for commercialization. My former boss's mentors, Len and Lee Herzberg, had two of the biggest patents at Stanford. They had the fluorescence activated cell sorter and then what are called humanized antibodies, which brought in hundreds and hundreds of millions of dollars to Stanford. And actually, they gave personally most of their own money away. They kept enough to survive, but then they gave most of the money away and they ran their own lab off of a lot of that money.